This recording is for Biology 1406. This is the fourth chapter and last chapter of Unit 4 on gene expression. The subject is viruses and viral genetics. Chapter 21 of the OpenStax textbook. All right. So a little bit about viruses. These are not living cells. They are subcellular structures. They are not alive. So they don't meet all the six typical criteria for a cell. You know, they're not a living organism. They don't reproduce on their own. They don't metabolize. They don't metabolize. They don't respond to their environment in real time, and a bunch of other things. They do, however, reproduce in a complicated way, and they show extreme organization, and they adapt over time. So they meet 3 out of the 6 criteria for living things, but they are not cells. All right. A universal thing, as far as we know about all viruses, is that they must infect or enter a host cell in order to make copies of the virus. That is a universal feature of everything that we've identified and called a virus so far. So they really are a parasite. They are a parasite of cells, and they utilize the cell's own metabolic machinery to make copies of themselves. The origin is really not at all agreed upon. There's some interesting theories. There are hypotheses that have been tested to various degrees, and there's support for a number of different competing hypotheses. So we'll leave that idea alone for the moment. One thing that's pretty consistent with viruses and gives us a little bit of information about their potential origins is the fact that their nucleic acids, their heritable material, because they do have DNA or RNA, tend to look like bits and pieces of the DNA and RNA and other living cells that have been grouped together. As a generalization, viruses are very specific to species, usually just one species, sometimes a couple, but that's relatively rare. The norm is that they are species specific, and even within those species, tissue specific. They only infect a particular type of tissue. For instance, the viruses that cause colds and flu and the novel coronavirus right now all infect epithelial tissue of the respiratory tract. That is their particular target, if you will, is epithelial tissue in a mucosal surface, which is your respiratory tract. All right. Some interesting experiments towards the end of the 19th century, so the 1890s here, very tail end of that 19th century, showed that there were very small particles. These were measured by using extraordinarily fine ceramic filters to separate materials that could be extracted out from ground up diseased leaves like these right here. And that extract that had been passed through an incredibly fine filter, which excluded cells, which excluded bacterial cells, and excluded a whole lot of things. The material, something got through the pores that were too small for even a bacterial cell to reinfect new leaves and transmit what we these days call the tobacco mosaic virus. At the time it was called tobacco mosaic disease. And it could be transmitted by liquid from one plant to another. And it turns out that something much smaller was the causative agent. We now know that that's the tobacco mosaic virus. In the 1930s, as it says right here, we had the first developments of electron microscopy, where you use a beam of electrons rather than a beam of light. Electrons in a beam have a wavelength, and it's a very short wavelength, much shorter than blue light. And as a result, you can make images of much smaller things because you get much higher resolution. So this is an electron microscope image of what turns out to be a bacteriophage virus attached to the surface of a bacterial cell. So just some examples right here. A virus particle, a viron, one virus, a number that is not unusual is about 20 nanometers in diameter, which is very small. Recall that your typical living cells, eukaryotic cells, are somewhere between 10 and 100 micrometers, and the smallest bacterial cells are down on the order of about 0.1 micrometer, or about 100 to 200 nanometers. So an order of magnitude bigger to 2 orders of magnitude bigger, or a factor of 2 in an order bigger than these things. All right. So electron microscopes have allowed us to actually see viruses, which was not possible before. So some scale right here. Here's the viral particle again. Here is a prokaryotic cell. Notice the scale bar right here, 50 nanometers. And then we jump out. So this surface right here of this prokaryotic cell would represent one of these things right here, these bacillus cells, adjacent to some eukaryotic cells. And notice the scale bar here, same size line, but now it's 1 micrometer. 50 nanometers is not 0.1, 100 nanometers would be 0.1 micrometer, but it is in fact 0.05 micrometers. 0.05 micrometers. Is that right? 100 is 0.1. Yeah, that's correct. All right, a little bit about viruses. There are some interesting ideas. The two general forms is that viruses are some sort of degenerate. That's where the word devolve comes from right here, degenerate. Things that are leftovers from some previous fully functioning cells. So are they something that, is derived from a once living cell? Another candidate explanation is that these are nucleic acid molecules that have escaped from cells. There is such a thing as a transposon, or a bit of genetic material that can jump from one location on one chromosome to another, or even from one chromosome to another chromosome within a cell. Humans have these, all animals have them, lots of plants have them. They're pretty widely distributed. They're called transposons, and they are bits of genetic material that can copy and paste themselves and replicate within a genome, and they're responsible for a lot of the genetic material in eukaryotic cells. And there's some molecular cues or bits of evidence that suggest that viruses may be some version of these transposons that succeeded in escaping the host cell, not just jumping from one chromosome to another within a cell or duplicating, but actually escaping the host cell and duplicating and introducing themselves into another nearby cell. That's a competing idea. Or that they are a self-replicating molecule of sorts that has become dependent on other living cells around and was never a part of a previous existing cell, neither as the escaped nucleic acids or a degenerate cell. Anyway, no answers for sure to this question. This is the life cycle, and I say life in parentheses right here because they're not really alive, of the novel COVID virus that is causing us to take this class online right now and causing so much upheaval and trouble globally right now. So it's called a coronavirus. Corona means, it refers to the pile of light, the wavering light around a star or sun. So that's where the name comes from, because under the microscope, under an electron microscope, they kind of look like little suns. And that corona is really the product of these things called spike proteins, which are proteins sticking out of the glycoprotein membrane surrounding this virus. So it's got these little spike proteins sticking out. And then, so there's a membrane, and then inside that there is a protein capsule. There's a nucleocapsid protein, so there's a bunch of proteins making up a capsule that contains the entire virus, but that's within the membrane. And then within that capsid, we're going to go ahead and have the genetic material, which in the case of the coronavirus is a single-stranded RNA. I believe that's right, single-stranded, yeah, single-stranded RNA. And so the way the virus works is the spike proteins bind to a receptor present on a number of our cells, including epithelial cells, called ACE2 receptors. and binding to that receptor gains the virus entry into the host cell. The receptor is brought in. This would be an example of receptor-mediated endocytosis. And here we see that the viral particle is within a membrane bubble. It's in a vesicle formed from the cell membrane of the cell that it's entered. The host, it is now its host. So what happens once it enters is that the virus is able to open up the encapsulating membrane by having that membrane fuse to its own membrane. So here we see the membranes looping back and forth, and that creates an opening, kind of like a nuclear pore, that allows its nuclear material to exit, and that's going to be this ribosomal molecule, this single strand of material, 5' to 3' RNA, and it's got a bunch of different genes on it. And what it does is it gets duplicated. So the complementary RNA strand is built, so it turns into double-stranded RNA right here. And then that RNA is translated to produce a whole bunch of different proteins. So that's the proteins. Some of those are going to include the spike proteins right here, the various other proteins making up the nucleocapsid, those proteins right there, and some enzymatic proteins having to do with manipulation of the RNA. So that's what all of these guys are here. And one of them is an enzyme called replicase right here. And replicase does something unusual, and it takes the viral RNA genome and writes a antisense version of it right here. So this is 5 to 3 right here. This is going to be 3 to 5 right here. It's in reverse because it's complementary, of course. And that is transcribed to make a whole bunch of new pieces of RNA right here, which are messenger RNAs that are going to be translated into additional proteins right here. So quite a complicated virus. It's got RNA nuclear material in here, some of which is used directly to generate some new proteins, including replicase, which creates an antisense strand of complementary RNA right here that is used as the template for transcription to make a whole bunch of new pieces of messenger RNA, and then all those messenger RNAs are going to be translated using the endoplasmic reticulum, ribosomes attached to the ER, rough ER, and then subsequent modifications of those proteins in the Golgi apparatus system. And using a chunk of membrane from the Golgi apparatus, plus the proteins produced right here that are bound to the membrane, we start seeing the assembly of a new viral particle right here with its spike proteins, its capsid proteins attached to its membrane right here, its ribosomal nuclear material here, and eventually we form a complete viral particle encapsulated in a vesicle formed from a piece of the Golgi apparatus right here, and by exocytosis, that vesicle fuses with the cell membrane, evaginates, because it fuses and folds inside out and throws out its contents. So the contents of the vesicle are released, and that's a viral particle right here. So that's the life cycle of the coronavirus, operating, as you can see, in the cytoplasm of its host cell, and all of its genetic material is in the form of RNA. All right, so now some general stuff about viruses. So here's an example of 3 different viruses. This is a bacteriophage right here, and bacteriophages are viruses that infect bacteria. So they don't infect eukaryotic cells, they're specific to bacteria. This is one of the most famous from a structural point of view bacteria, because people who look at it, and this drawing is somewhat stylized right here to kind of accentuate the almost machine-like quality of the virus, but that's kind of what they look like. This electron micrograph is where that imagery comes from, this thing right here. This thing right here. So you see these little filamentous tail things right here, some protein structures that allow it to attach to a cell wall and punch through it, a helical structure right here, and then a isocohedral capsid head right here, and then the genetic material will be contained within that. All right, so just as an example. All right. And so this is a virus that consists of DNA, its nuclear material, and then a bunch of proteins making up the outside of it, and the proteins that do things and protect the DNA inside. Here's an adenovirus, which is not too terribly different from, which is kind of similar to our cold viruses. This is creates another respiratory disease right here. It's got DNA as its nuclear material. So nothing terribly unusual right there. It has a protein package surrounding the entire thing. This is a capsid, so it's got this protein capsid right here. And then it has these glycoproteins sticking out from the surface, which bind to receptors on host cells and allow this structure to gain entry to that host cell via these signal molecules binding to receptors on the target. Here, very much like our coronavirus, is another encapsulated virus. So it's got a cell membrane, as it were, around the perimeter here, and that membrane is derived from its, partially derived from its host, from the host that the cell emerged out of. So this membrane and a bunch of its proteins on the surface of the membrane are derived from that host, which means it can infect that host or that host cell or other closely related cells. Internally, there's going to be a capsid container made out of proteins. That's quite normal. And then inside that, in the case of the HIV retrovirus, RNA and ready-to-go enzymes of reverse transcriptase, which can take RNA and rewrite it backwards into DNA. This is something that does not normally happen in normal cells. Interestingly, there are some transposons called retrotransposons that the transposon is transcribed as RNA, and then that RNA is retrotranscribed back into DNA in a new location, and that's how those things propagate. So that's one of the reasons why escaped nuclear material in viruses are sometimes considered to be the same thing. But anyway, so this virus right here has a protein outer coating and nuclear material inside, which happens to be DNA in this case. This virus here has a protein outer container. This is all viruses have that proteinaceous external container, and then some nuclear material inside. In this particular case, it's DNA again. Here, we've still got that protein capsule containing the nuclear material. In this particular case, it's RNA and reverse transcriptase, but this virus is also surrounded by a membrane. So at bare minimum, the viruses, what you need to know for a test is that all viruses have nuclear material and then a surrounding protein structure that provides protection, as well as the mechanism, whatever it is, for getting into the host cell. This one's just an aggressive puncher through the host cell. This thing tricks the host cell into taking it in because of these glycoproteins stimulate receptors on the host cell. Same thing for these spike-like proteins right here, that these glycoproteins stimulate receptors on the host cell, and that cause that cell to go ahead and pull the viral particle in. So the envelope is the variable bit that may or may not be present on viruses. All viruses have nuclear material surrounded by protein. A little bit about shapes. A lot are geometric, like this icosahedral shape right here, which we could count this up, but I'm not going to bother with it. I think it's 12 sides, but however many it is right there. We can have filamentous viruses, long spiral helixes. This is a characteristic of the tobacco mosaic virus. A lot of plant viruses look like this. A lot of animal viruses look like this. And then the enveloped viruses, where this is simply a membrane surrounding what would be, something like this, for instance. And then finally, the head and tail viruses, which are characteristic of bacteriophages, viruses that attack bacterial cells. So there's the head and there's the tail. All right. So let's take a closer look at an example of a bacteria to learn about bacterial behavior. So this is bacteriophage T4, bacteriophage T4. Again, it has a nuclear DNA material. This head capsule right here, which is an icosahedral capsule containing the nuclear material, and then a pretty complex protein tail with tail fibers and attachment points and all of this. And its target is going to be the bacteria E. coli. So the adenovirus here, just looking at it a little more closely, nuclear material in the inside, protein around the outside right here. In this particular case, this is a adenovirus that infects the respiratory tract. It's one of the other sources of colds, aside from coronaviruses, which also cause colds. It is non-enveloped, and as it says here, there's some related viruses. Poliovirus, which is quite harsh on people. Human papillomavirus, that's HPV, the virus that causes cervical cancer and rarely reproductive cancers in men as well. We've got a vaccine now that's made this hopefully a thing of the past. And the hepatitis A virus. Hepatitis, there's 3 different viral types, A, B, and C. A and B are relatively treatable. C is the long-lasting chronic one that is so difficult to get rid of once that viral infection takes hold. All right. We're looking at the HIV retrovirus. Of course, HIV is the virus, human immunodeficiency virus, that causes AIDS, acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. HIV causes AIDS. In the language of the current viral pandemic, the novel coronavirus or COVID, corona SARS-6, I forget what it's called, is the virus, and then COVID-19 is the infection, the name for the active infection caused by the virus. All right. So viruses do have genomes. They've got, a pile of DNA, and all the DNA of a thing, normally of a living cell, is called a genome. So viruses also contain a genome, all the DNA they need or RNA they need to get stuff done. Right, so the core of all viruses has nucleic acid, either DNA or RNA, but never both so far. Either that DNA or that RNA can be single-stranded or double-stranded. They come in both varieties, right? More commonly, the DNA viruses are double-stranded because viruses genetically look like their hosts. And all living things out there, their DNA is double-stranded. So most of the the DNA viruses are double-stranded. And similarly, most of the RNA in most cells is single-stranded. Think of messenger RNA or transfer RNA or ribosomal RNA. It's only one strand. It may be folded and bound to itself, but it's not folded and bound to a complementary strand. And so since that RNA is normally single-stranded, we'll see that single-stranded is typical for RNA viruses. They're typically single-stranded RNA viruses, typically double-stranded DNA viruses. Those are your 2 general choices out there. Also, like their hosts, viruses that infect eukaryotic cells generally have linear genomes. Their DNA or RNA is a linear piece with a beginning and an end. And for the viruses infecting bacteria, their nuclear material, whatever it is typically circular, like a prokaryotic genome. And also those genomes, all of that DNA or RNA is going to be far, far less than what you would find inside that cell. Even a bacterial cell has orders of magnitude more DNA in its genome than most viruses. So viruses are they just have a few key genes for a few key proteins that allow them to take over the host cell, some key proteins that allow them to enter the host cell, and some key proteins to form the capsid that protects the DNA while they're outside of their host. Yeah, and then we'll go ahead and take a look at the mechanics of a DNA virus versus an RNA virus. One interesting thing is RNA does not have the editing and repair mechanism that DNA does in host cells. And so therefore when RNA is made, if there's an error, the copy is not exact, there's no proofreading and no repair mechanism to look for that error, look for that mismatch, and repair the new piece of RNA to match the old piece of RNA, like there is in DNA. That's pretty routine. And viruses don't, proofread and check their DNA, but the host cell proofreads and checks the viral DNA if it's, if it is DNA that makes up their genome. And so, anyway, the long and short is that DNA viruses tend to evolve slowly. RNA viruses mutate quite rapidly, which means they evolve quite rapidly. They mutate into versions that don't work; those get eliminated. But occasionally, those mutations make them more infectious or more create greater symptoms, so that the person's more likely to transmit the virus to another person. Coughing and various other secondary effects. Anyway, so RNA viruses are typically more rapidly adapting to their environment, and these tend to be the ones that are more dangerous and more difficult for us medically. All right. We can classify viruses in lots of different ways. I'm not going to worry about that. What I do need you to know is what kind of nucleic acid types are out there. So remember, double-stranded DNA, quite common. Single-stranded DNA, those exist, but they're pretty rare. Double-stranded RNA viruses, pretty rare, but they do exist. Single-stranded RNA viruses, lots and lots and lots of them. DNA viruses inevitably interact with the nucleus of the host cell, which has some very interesting implications. RNA viruses typically only work in the cytoplasm of the host cell, so they're in and out quickly, just manipulating cytoplasmic processes of an existing cell. The exception to that is going to be retroviruses, where RNA is rewritten backwards into DNA, and then that DNA interacts with the host cell's nucleus. All right, so here's a little bit about that and some examples of viruses that have those particular structures. So, two. Yeah. All right. We're not going to worry about identifying them via their capsids, what the heck capsid looks like. Just that they have them. All viruses have a DNA or RNA core and then a surrounding capsid, a capsule that they're in, made out of proteins. This is the most widely used classification system right here, and it has to do with their nuclear material and how messenger RNA is formed, right? So in this particular case, a double-stranded DNA, the messenger RNA is made directly from DNA. Single-stranded DNA is converted to double-stranded, then you make your RNA from that. Double-stranded RNA is used to transcribe fresh RNA. Single-stranded RNA plus, this stuff acts as the messenger RNA. The inverse right here, this single-stranded negative sense, the messenger RNA is transcribed from the RNA genome. So we read this piece of RNA to make messenger RNA. And then the single-stranded RNA virus with reverse transcriptase that rewrites itself from RNA back into DNA. That's the HIV virus right here. And then these curious viruses right here, where they are DNA. The DNA is rewritten as messenger RNA, or transcribed to make an RNA intermediate. But then they also use reverse transcriptase to take that RNA intermediate and rewrite back into DNA again. So pretty complicated life cycle, even for a virus right there. You can go ahead and take a look at this presentation right here, which will illustrate categorization of viruses. Let's go ahead and take a look at this little video. Well, I can't get that animation to play, so we'll just have to bypass it. But if you can make it work on your own PowerPoint, do so, and take a look at that animation. All right, cytopathic effects. So why do we get sick when a virus, is in us? The virus itself oftentimes does not cause any harm. Kind of depends on the virus. At the bare minimum, in a lot of viral infections, especially severe quick infections that come and go, like the flu or a cold or things like that, what happens is a lot of the infected cells are burst and killed. They're destroyed. Lysis, the cell is ruptured. Basically, it fills up with viral particles and then bursts, releasing the particles all over the place. If enough of your cells have this happen at the same time, it's going to cause you functional problems in your respiratory tract. Additionally, a lot of cells in a lot of infections are induced to kill themselves, in order to stop the infection. But the infection runs through those cells quite quickly. And so even though programmed cell death as a means of protecting the greater body is quite normal in us, if enough of this happens in enough tissue, especially somewhere sensitive like the respiratory tract, we're going to have problems and feel the effects. But typically what makes you sick is your immune system. It's the chemical warfare that your immune system launches on the infected cells and the viral particles themselves if it can make contact with them. And the greater the stimulus by the infecting agent, the more of an alarm basically that the virus induces, the more violent the immune response is and the sicker you're going to feel while that immune response is going. And sometimes the immune system becomes so involved in attempting to wipe out a viral or bacterial infection. That's a separate thing. But the idea is similar. Immune system marshaling resources to get rid of an infection. That it occupies so much of the body at the same time that the chemical warfare that is so much a part of the immune response is destroying the body as it's trying to save it. And that's what routinely kills people with the flu. And with the COVID-19 infection is that the immune reaction is so substantial. It's kind of like a version of anaphylaxis, which is a body-wide hyperimmune response to an allergen. And everybody knows anaphylaxis is theoretically fatal. Well, it's a variation of that same thing that's happening in these extreme immune responses. Now, there are some viruses, HIV is one of these, that leaves a cell by budding out of its host cell and therefore does not kill the cell. The coronaviruses do the same thing. They don't automatically kill their host cell. All right, so what has to happen in all viral infections? We've got to attach to the target cell, the host. We've got to get in there one way or another. We've got to replicate all the pieces, which at the bare minimum is the proteins of the capsid, plus either the DNA or RNA that makes up the genome of the virus, and then all those pieces have to be put together. The capsid has to be assembled around the DNA or RNA, and then that thing's got to get out of the cell or be released from the cell by the host cell or something. All right, let's talk about attachment. So the key thing is receptors. Most viruses attach only to a target cell that has the right receptor molecule. That's why viruses are so species specific, because one way to keep one species separate from another one is by having different versions of the same receptor for a similar function on cells. For instance, eggs and sperm cells have receptors that allow conspecific sperm to bind to and ultimately penetrate and fertilize a conspecific egg. Those receptor molecules are different enough from one species to another that the sperm from one closely related species usually can't bind to and fertilize the egg of another species. Same with the viruses here as far as specificity of receptors. So that makes viruses species specific, and often for the same reasons. Some receptors are only present on certain cells, tissue specific. As we kind of saw before, but just to get it down on paper clearly right here, bacteriophages punch a hole through the cell wall of bacteria and then introduce their nuclear material directly into the host cell, leaving the entire capsid structure outside. The viruses that infect eukaryotic cells almost always bind to the receptor and then gain access to the membranes and either fuse their membrane to the host cell's membrane, and then the capsid plus its nuclear material enters the cell that way, or their receptors allow them to induce invagination through the cell itself, receptor-mediated endocytosis, and the cell is brought into the host cell in its own capsule made out of membrane from the host right here. Excuse me. So, generally speaking, replication and assembly. What does a DNA virus do? Well, the standard answer is, you're going to duplicate the DNA, to make a new virus. So that hijacks the DNA copying apparatus of the host cell. Plus, to make the capsid for the protein for the viral virus, we need to transcribe some of the genes on the virus genome into messenger RNA to make those capsid proteins. In RNA molecules, depending on the details, the RNA molecule is used as the messenger RNA or as the inverse mirror image of the molecule, and then we copy the RNA into proper messenger RNA to make all of the various proteins right here, and we also copy that RNA directly to make a fresh viral genome. So, one way or another, we're using this both as the template for copying and the transcriptional template for translating. And then RNA retroviruses, by far the most, really the most dangerous and the most complicated, and in a lot of ways the most interesting viruses. That RNA is rewritten backwards into DNA, and then DNA is used both as a way of synthesizing fresh messenger RNA to make the capsid proteins, and then that RNA is also going to be duplicated. And in DNA viruses, both types here, but particularly the retroviruses, the DNA can actually be incorporated into the host genome, so planted in the host genome and left there as a piece of DNA. So that's a very interesting thing right there. Any DNA virus, in theory, has the ability to do that, or virus that, via retro functions, can go from RNA to the DNA state. All right, so again, release of the viral particles either can be massive and rapid and kill the host cell in the process, or it might include budding right here, which is not necessarily lethal, not instantly lethal to the host cell. So here we're looking at an influenza virus. It's an encapsulated virus right here, kind of related to the coronaviruses. It induces the host cell to invaginate, receptor-mediated endocytosis and take in the viral particle surrounded by some membrane from the host cell. Eventually, that structure disintegrates itself, and the nuclear material is, left free out here. The viral RNA enters the nucleus, where it's replicated by RNA polymerase to make lots and lots of copies right here. That RNA is also used as a template for translation. So we make the the capsid particles, the replacement capsid particles right here. There's the viral proteins. And then all of that material is assembled back together. Fresh RNA nuclear material, plus capsid, and exocytosed out of the host cell. So this happens in eukaryotic cells as well, but it's most well understood in prokaryotic cells, and that is the two general life cycles that any virus can go through. In this case, we're using as an example a bacteriophage, which is a virus infecting a bacterial cell, but this also works for eukaryotic cells. There are two life cycles, the lytic cycle, you've got to know this, and the lysogenic cycle. So the lytic cycle is the one where a virus infects a host cell, nuclear material gets injected in here, the phage DNA, and it's used as a template to make lots and lots more DNA, and as a template to make RNA to be translated to make proteins, so it can make the capsid proteins. Here we're making some fresh capsids right here. And that's done very, very rapidly in the lytic cycle, so that we make lots and lots of viral particles quite quickly inside the host cell, using the host cell's resources and all of its genetic equipment to duplicate these viruses, and then in the end, they explosively leave the cell, usually just by rupturing the cell because it's overfull of these viruses right here, and so the host cell dies. The lysogenic cycle, on the other hand, is far more sinister, if you will, and far more interesting. So the genomic material is still introduced into the host cell, but instead of immediately being transcribed and copied, the genome is put back, as it were, taken, reintroduced into the nucleus in a eukaryotic cell, or brought back to the nucleoid region of this prokaryote, and put back where the cell thinks it should go. The repair enzymes that handle keeping DNA intact. There should be no loose ends on a bacterial chromosome. It should be a circle, so when they find a linear piece, they're going to try and fix it. And they fix it by putting it back somewhere in the genome of the host cell. Well, now the virus is really asleep. It's a zombie. It's just being carried by the host cell passively. When the host cell reproduces and divides to make 2 new cells, now we've got 2 copies of the genome of the virus. And this can happen again and again and again until we have a huge population of bacteria that are carrying the virus but aren't actually infectious at the moment because the virus is in a dormant state, just being carried as genetic cargo, one of the many genes that faithfully gets copied but doesn't get translated normally. And then under stressful conditions, like when these host cells are having difficulty, getting resources, or other mechanisms that let the basically let the virus know that the cell it's in is going to be in close proximity to other cells or is in danger of dying. Viruses in the lysogenic cycle can switch back over to the lytic cycle and escape their current host and try and jump to another host. That's what the lytic cycle is for, is from getting from one host to another. The lysogenic cycle ensures long life of the virus within its host. In a multicellular organism, it'll inhabit cells for a really, really long time. And as those cells are replicated, it's replicated as well. In the case of prokaryotes right here, it can expand through a population as the population expands. So whichever thing allows the virus to continue existing for generations of time is what works, and natural selection takes care of, deciding which of these works. Natural selection has favored this for an awful lot of viruses, but it's also favored this for a number of viruses, particularly in eukaryotes. All right. So there's a bunch of different animal viruses. You don't need to memorize any particular ones right here, but there's plenty of them. All right. There are also, we'll take a look at one interesting one here just to talk about some things that these viruses do. So as you see right here, varicella zoster is the virus that causes chickenpox. It's enveloped, right? So it's got a membrane surrounding an icosahedral capsid, and it's going to have DNA. Its double-stranded DNA genome becomes incorporated into the host DNA. Right, so this is a virus that infects eukaryotic cells, and its genome can be put into the host cell and become dormant. This life cycle right here. And so the cells in you or me, because humans are a target of that virus, can carry this DNA for years and years and years and years. We have the initial infection, chickenpox, that a child gets. Smallpox is a relation of this, but happily gone from the human population now. But you never really lose the virus. You get over the initial chickenpox infection. There it is. Some of the you'll get whelps like this all over your body and swelling and all kinds of things going on. But the infection can return in the form of shingles right here. And so this is an eruption, and what's happening is the virus was sitting like this in nerve cells. It infects cells of your central nervous system, sensory cells to be specific. And so it's sitting in the nucleus of those sensory cells. And then periodically, it switches from the lysogenic cycle back to the lytic cycle right here, mass produces a bunch of viral particles which are transported down the neuron. They don't kill the neuron, but at the end of the neuron, where the dendrites are close to the surface of the skin, the viral particles cause an inflammation as they're ejected from the nerve cells, and this is how this infection can get from one person over to another person. Same thing for the herpes simplex viruses. They've got a very similar sort of life cycle, and that's why they last for the duration of somebody's life. All right, here we're looking at the HPV virus right here, human papilloma virus, I'm sorry. As you can see right here, it's just a capsid, an icosahedral capsid, so no membrane around it, just the capsid itself, and then there's going to be that DNA inside. And this is a cancer-causing virus. It leads to cervical cancer because the DNA is included in the host's DNA. It messes up the expression of genes in the host. That's where the cancer comes from, damaging the host's DNA when the viral DNA is included in the host's DNA. There are quite a few plant viruses. We first learned about viruses by studying plant diseases, and a little bit about how they're propagated. Horizontal transmission is the movement of something from one individual to another in the same generation. So, when you catch a cold, you're getting it by a horizontal transmission. Vertical transmission is, down generations. So, for instance, in humans, there is HIV can be passed on from a infected mother through the placenta or during childbirth at the very latest, to the baby, the embryo or the baby. So a baby can be born with an HIV infection. That would be an example of vertical transmission. All right. You don't need to remember anything about plant viruses. Just know that they exist, and like animal viruses, they can be DNA or RNA. And there are quite a few different, varieties of these viruses. Impacts on, humans. Take your pick. There's a virus for almost every organ system and every bit of our body somewhere. Vaccines. Very, very important. So what vaccines do, of course, is trigger the latent immunity that we have. Theoretically, our immune system can make us immune to anything. It shouldn't make us immune to anything that normally occurs in our own body. When that guard fails, that's what we call a autoimmune disorder, where your own immune system attacks something in you that shouldn't normally happen. But you should, in theory, be immune to everything theoretically out there in the environment. Now, there's some feedback mechanisms that make you not immune to food. And when that malfunctions, we call those food allergies, because you become immune to that food. And when you get it in you, you have a big immune reaction, and so on and so forth. Anyway, how can we trigger the immune response? Well, that's where we take something that mimics the antigen. Antigen is something that creates an antibody response in an immune system. So an antigen is something on the surface of a bacteria, something on the surface of a virus, something on the surface of a nut, for people who are allergic to nuts. All of those are antigens. But we can take some antigens, either from a live virus right here, live being in parentheses because they're not really alive, but a virus that's been damaged, attenuated, so it's not particularly infectious anymore. But you can introduce this non-replicating, non-infectious virus into a host cell, into a host by injection here into a person, and the immune system is going to recognize all the bits and pieces of these viruses and create a whole bunch of antibodies and generate a remembered immunity to that virus. Same thing can be done with like heat sterilized viruses where key proteins are still present, but the nuclear material's been disabled. Or we can take just little fractions, bits and pieces of the viral particle, bits of DNA, but typically it's going to be bits and pieces of their capsid proteins, the things that your cells would normally see when they encountered a virus, an intact virus. It's going to be something on the outside of the virus. But all of these can be used for immunization. And notice even the attenuated live viruses is very, very low risk of infection. When people have a really, really terrific violent response to immunization, which a few people always do, when you're talking millions or hundreds of millions of people being vaccinated, there's always going to be a couple that have an extreme response. A lot of times it is that they're hypersensitive to the immunogenic, components of this virus. Those are the same people that would probably die, have even from a normally non-lethal viral infection. So these people are, no matter what, however it comes, they're going to have an overly aggressive response in their immune system, and that, basically their immune system goes wild and destroys their body. That's very rare. It's an extremely rare thing, and it's a necessary price to pay for what is normally an extraordinarily effective medical technique, despite a lot of people right now resisting viruses. Generally, they are scientifically illiterate or have been misinformed terribly or have some sort of other set of criteria that is more important to them than the mechanism of the vaccines. But usually there's a lack of understanding of how vaccines work, which leads to people rejecting the idea of those vaccines. So here's an example of the HIV virus infecting a host cell right here. And the important thing is, of course, is that it's an RNA virus, but it also includes reverse transcriptase. And here we're talking about ways in which you can fight the virus. So, inhibitors that inhibit reverse transcriptase is one of the tools used to combat HIV infections. But anyway, the viral particle binds to a receptor on a helper T cell. This is a cell of your immune system. The viral particle itself, with its capsid and its RNA, is introduced into the host cell. The RNA is rewritten backwards as DNA and included in the genome of the host cell, and it can go quiet in here. It can sit in here for years in these long-lived helper T cells, and that would be the lysogenic cycle. And some of these helper T cells are capable of replicating, and when they replicate the viral genome as well. And then at some point, usually when this cell is under stress, when the host is under stress and the immune system is weakened, stress weakens immunity, unfortunately, then the lysogenic cycle switches back over to the lytic cycle. Viral particles are produced and emergent viral particles are butted off of the host cell right here to infect other helper T cells in the immune system. By HIV infecting helper T cells, it does two things: reduces the number of helper T cells in the body, and also causes them to malfunction, so they don't carry on their key immune functions. And helper T cells organize and direct the immune response of the host of those helper T cells. So if you knock out the helper T cells, the entire immune system loses its focus and its ability to identify threats and respond to them in a massive way. And that's the acquired immunodeficiency syndrome, AIDS. All right, a few other things to wrap up this presentation. We're just about done. Prions. These are not viruses, but there's no good place to put them, so we put them over here with viruses in the chapter on viruses, because that's more of a correct place. These are not living things, but these are proteins that are infectious, right? So there's three things you can get infected from: bacteria, viruses, and certain proteins that are infectious, right? As it says right here, they're very small, even smaller than viruses. They don't have any DNA or RNA, so there's no heritable material in them. And they cause a number of really quite fantastic, from a medical point of view or a scientific point of view, and quite terrible from a human perspective, diseases right here. Mad cow disease, which most of y'all will be familiar with. It occurs in cows, bovines. The human version of it, which you get if you eat infected nervous tissue from a cow, you get the human version of it called Creutzfeldt-Jakob's disease, which does the same thing. It's mad human disease, I suppose, because it destroys the human brain. Kuru, another version that is formed in humans and transmitted from one human to another, classically by by eating the tissues of another human being, and it's nervous tissue again. A variation of mad cow disease called scrapie from sheep, and it's well understood that scrapie is the source of mad cow disease, and mad cow disease in turn is the source of Creutzfeldt-Jakob disease. So these three are all related to each other. Kuru is a completely distinct version of prion disease that was identified in Samoa and New Guinea, Papua New Guinea particularly, and some of the other Southwest Pacific islands, where unfortunately this habit of ritual, it's never for actual sustenance, that could never work, but ritual, for symbolic reasons, cannibalistic activity has long appeared. And then there's a more recently discovered one, the last few decades, chronic wasting disease that we see in a number of different animals, including deer, and elk and moose are just very, very big deer, and then deer. These are all really closely related species right here. And one of the interesting things is prions are not destroyed by cooking. Viruses are killed by cooking. Bacteria are routinely killed by cooking. So cooking food normally protects us, but you cannot get rid of the infectious element in any of these diseases by cooking. It remains intact and functional. So here's an illustration of what's going on. The prion is a misfolded version of an endogenous. We don't need to worry about these statements right here, but a superficial protein associated with nervous tissue. It's a membrane-bound protein on neurons in the central nervous system of mammals. There's also some versions of these proteins in birds. So birds and mammals carry variations of these proteins. No other animals do. And as far as we know, prions only affect mammals and birds. And it's because of this related set of very, very, very complicated enzymatic proteins associated with the surface of neurons in both the distal, the peripheral nervous system, so the sensory nervous system, or the motor neurons, and in the central nervous system of the brain. So these proteins can fold into an incorrect shape, which is represented here in prion PSC right here, the incorrect shape right here. All right, that's one way in which prion disease occurs. And then what's really interesting is when the protein gets folded into the incorrect shape right here, if it touches one of the regular proteins that's in its normal shape, it induces that protein to reshape into the infectious form. That A, doesn't do its job, it doesn't work anymore in nervous tissue, but it can propagate by folding these proteins, which are abundant in all of the nervous tissue of the host, converting them from their normal form into this malfunctioning version, which in a chain reaction, as one touches another, each one in turn gets changed. And what happens is this infection inevitably starts in some peripheral nervous tissue, usually of the digestive tract. You've eaten some contaminated meat. Nervous tissue, it has to be contaminated nervous tissue, of a host organism, another human, a deer, an elk, a sheep, a cow, with the version of its protein. And then that contaminated tissue has one of these prion particles, and if in your digestive tract, in your stomach or your small intestines, even one of these proteins is exposed on the inside your digestive tract, you've got a little teeny bit of damage to the epithelium, and the tip of a neuron of a nerve is in contact with the fluid of your digestive system. Think of an ulcer. That's what's happening right there. The prion can then touch that protein, change it to the prion form, and then that will do it to the next one and the next one and the next one until all of the proteins are converted into the prion form. When enough of this happens, we destroy central nervous tissue. And that neuron in the digestive tract leads back, inevitably, all the way back to the central nervous system and all the way to the brain. So given time, and it'll take a few years, the prion infection will work its way all the way to the brain. It's not that the particles are moving, it's just a chain reaction of one particle inducing the change in the next one until we reach the central nervous system. Then it spreads out to through the central nervous system, the brain and spinal cord, until at some point it becomes fatal. And inevitably these are fatal. And that's prion disease. One last little bit of information here. There are small infectious pieces of just nuclear material that don't have any proteins. These are called viroids, which just means a very small virus. They seem to be all RNA so far. No protein capsid around them, and these are unique to plants. There's no analog in animals that we've ever found. They don't manufacture any proteins, but what happens is the little chunk of RNA gets replicated, and if they induce a lysogenic reaction in the host cell. Let's go back and look at those life cycles really quickly. the lytic cycle in the host cells, mass producing lots and lots and lots of particles right here, we end up with an explosion of just those little rings of RNA leaving the host plant cell, killing that cell, and if that happens to enough cells at the same time, we do great damage to the plant. When we were talking about shingles, chickenpox, herpes viruses, and even HIV right here, all of these are viruses that are included in the genome of the host cell. So that's that lysogenic cycle right there, where they're cryptic. Anyway, prions are neither of these nor of these, and viroids work like this and replicate very, very rapidly, but it's only nuclear material going in, nuclear material coming out, and so far they all appear to be RNA. And that is it. That is the conclusion of that chapter in this unit.